All right, everyone, thanks so much for joining us tonight for our second installment of our Missouri Species Academy Creek course on an introduction to mussels. And tonight we'll be discussing a little bit about mussel ecology, distribution, and how to collect them, and some of the things that go along with that as well. First of all, again, thank you for joining us. We are part of the Missouri Stream Team program, and I'm Amy Meyer. I'm with the Missouri Department of Conservation, and I help coordinate stream teams in the Missouri River Unit. And with us, we have Lacey and Jenna Steek as hosts as well. They are also part of the Missouri Stream Team program. April is our volunteer water quality monitoring coordinator, and Jenna is our stream team biologist that coordinates stream teams in the southeast region of the state, and also works in water quality monitoring. We also have Sam Doherty, our water quality assistant. Tonight, we're going to have Travis Moore, a fisheries management biologist, coming to us from Hannibal, Missouri. And a little bit about Travis. He is a fisheries management biologist. He is out of Hannibal in the Northeast region. He's been with MDC for 30 years, so that's awesome. Working on a variety of aquatic projects, including both private and public land fisheries management, big rivers and lake sturgeon management, and aquatic outreach efforts, in which we'll have a presentation on that coming up on September 18th, so stay tuned for that. His freshwater mussel experience has been on interior streams of the Northeast region and the upper Mississippi River. So with that, I will go ahead and pass the ball so that, oh, I can get you to show up. Thank you for being patient with us. Oh, chat. Okay, here we go. All right, so now Travis, you should be able to hopefully share your screen.
Okay.
And I'll go ahead and turn it over to you.
Okay. So can we see the Steve's mussel slide then?
Not yet. I cannot yet.
Okay. Let's see. Where it said that I was the presenter now. Let's see if it...
And again, everybody, we thank you for your patience.
Here we go. Here we go.
Great.
Okay. See something happening now.
Yes.
From this slide. All right. So as Amy said, I'm a fishery biologist up in the Northeast region. This presentation was actually supposed to be done by Steve McMurray, who is our MDC Mussel Ecologist. And Steve had a family issue come up, and he was debating about how best to try to do this. We really didn't want to cancel it or reschedule it. And I told him, I said, you know what? I said, be willing to do it if you'd like. So you're going to see Steve's presentation given by Travis. I won't say that you won't have a little bit of me mixed in, but it's going to be Steve's presentation that you're going to end up seeing. So I'm going to try to offer pretty much the way that he normally does for these events. Okay, I get this fixed right here. Pull this off to the side. Okay. So Steve's going to talk a little bit about just general mussel ecology, how freshwater mussels fit into our aquatic ecosystems, where they live, what are some of the factors that affect their distribution, their populations across Missouri. There are several of them that have adapted to be either habitat or host specialists. And those are some unique circumstances. And then we'll wrap up with talking about some collection methods, where you can find mussels and how you can find them. And those last three are kind of important because in a couple of weeks, Steve's going to give a presentation on mussel identification. And we have an assignment that we're going to toss out to you at the end of this presentation. So when we get to the collection methods and how to find the mussels, pay close attention, and I'll give you that assignment at the end of the presentation. So mussels really are aquatic natural biological filters. They process a lot of water, pull a lot of sediment and other material out of the water. And so in a lot of our streams, they're really working to help clarify that water. And then they're making those materials that are being pulled out of that water more usable for some other animals. And we'll talk about that a little bit later. They can exceed all other benthic biomass in our river systems, simply because of their numbers and their sizes, their mass. You can see from the photos that he's got here, they can get to be pretty big. And in the case of the picture of the spectacle case down at the bottom, there's just so many of them packed into those areas that they just take up so much of the biomass within that system. Important food for a variety of different animals out there. For muskrats, they're vitally important. Muskrats are probably the #1 predator on freshwater mussels. But there are also a couple of species of fish, like a blue catfish and freshwater drum, that prey on them too. And then they are indicators of good water quality. In many ways, they are our canary in the coal mine from an aquatic perspective, and they give us a pretty good indication about what's happening in those streams. And finally, they have tremendous effects on the other critters, especially the other benthic critters that are in our stream systems. They provide substrate for some animals to live. They provide places for periphyton and algae and all those different things to be and add to that community structure. Now, this is a busy slide, but there are a couple of things that Steve wanted me to point out here, and several of them are related. So as freshwater mussels are out there and they're feeding, filter feeding in that system, they're pulling water in, they're sorting through the material that they take in. And if it's food particles that they can use, then they'll go ahead and digest those items. But if it's material that they don't need, then that gets packaged up into the pseudo-feces packets, which are then extruded out into the rest of the system. And so what they're doing is they're making some of those materials that are dispersed a little more compacted into these little packets, these false poop packets. And there are things like crayfish and some insects that makes it easier for them to get and to digest those items. And I kind of liken it to, you could go out there and you could try to find an individual potato chip, where maybe you go out and try to collect an entire bag full of potato chips. And that's kind of what this is doing, is it's making those food particles more easily accessible to some of those other animals. The mussel shells provide habitat when they're alive. There's other things that live on the outside of the shell. And then even when that mussel dies, that shell is providing habitat for juvenile mussels that are out there. There are some species of catfish called mad toms that use them, just a variety of different things. And then there's also this bioturbation of sediments. And as the mussels are out there, many of them do move around. And as they're doing that, they're loosening up the sediments that are out there, whether it be sand or gravel, keeping those broken up so that some of those animals that need to burrow and use those other spaces can still have access to them. Okay. All right, so in Missouri, you're most likely going to find mussels in our stream systems. There are a few that have adapted to live in lakes and reservoirs. There's even a species or two that have adapted specifically to live in wetlands. But by and large, our streams are where you're going to find them. And here is a slide that kind of shows some of the places that we have sampled over time. And you can see southern Missouri is just riddled with dots where we have found mussels. And as you work into my part of the state up in the Northeast region, we have quite a few in our river systems up here as well. But you'll notice that in that northwestern third of Missouri, there just isn't much. And there's probably a couple of reasons for that. One is you start to look at where the glaciers came across Missouri, and that kind of marks about where the boundary was, where it came into the state. But the other thing is, I grew up in that part of Missouri, and I can tell you the stream systems up there have been severely altered. You can see lots of straight lines when you look at the maps and see how all of those streams have been channelized. And so much of what could have been good mussel habitat was destroyed back in the early 1900s and even prior to that. But as we break down these river systems into smaller units and really start to look at, where do we have the diversity of mussels within the state? And if you've been out, floated some of these streams, you're going to see some places that are very familiar to you. You're going to see some streams like the Black, the Merrimack, the Burbus, Gasconade, Osage, Big River. Those places have high diversity in them. And even as you come into Northeast Missouri, you start to get into some of the Salt River Basin, and some of those systems as well have some pretty good diversity. And although it's a little hard to see on here, if you include the Mississippi River in that, if you go from St. Louis up to the Iowa border, we have a tremendous amount of diversity in that system as well. We'd be in that 30 to 42 category. But that really changes once you get south of St. Louis and start getting into the open river. The habitat changes, and it's just not as stable, and so we don't see the mussel resource there like we do in the pools. Steve was working with staff from the University of Missouri Co-op and Tennessee Tech Co-op units, and they were looking specifically at trying to develop a model to predict where we should find mussels if we were to look at a particular stream prior to going out. And they used the Merrimack River system because we already knew where a bunch of the mussels were in some of those locations. And they could use those sites to look at a variety of different parameters and see what led to those places being more likely to have mussels. And so what they discovered is there were 4 specific parameters that tended to relate with mussels. One was a proximity to a bluff, if that habitat was nearby. Distance to permanent gravel, stable gravel. If the stream channel was laterally stable, and then was there access to water during low water periods? Again, mussels got to have water to survive. And so when we have those drought periods, we need some permanent water for them to be in. So if you look at the diagram that he has up in the upper right-hand corner, start looking at this system, and in this section, reds and oranges are your hot zones. Those are the places where you're going to more likely to find mussels. And you start getting into the blues and greens, that's going to be the opposite. You're less likely to find something in those sites. And so they're able to use this to kind of predict and help us be a little more focused with our sampling efforts. So here's a reach of stream where there is a known mussel bed. And one of the things that those of us that work with mussels, we sometimes get into some interesting discussions and debates about what constitutes a mussel bed. And there are different parameters that some folks use. For instance, some people will look at how dense is that mussel bed? How many mussels are in a square meter, for instance? And they may say, well, there needs to be at least 10 in this 3x3 area for that to be considered a bed. But for what I see in the Mississippi River, that's not necessarily the case. I might look a little more at how much area overall that those mussels cover, or I might look at diversity. Do I find a certain number of species? State of Illinois, for instance, they have to have, I believe it's 12 species in a bed in order for it to get some special status for some survey work and things like that. So a bed can be short, it can be long, it can vary a lot, but there's a lot of variation even within the bed. And in this particular photo, we're going to flip the color schemes. And what Steve did is they did some transects on this known mussel bed, to see where they found the most mussels. And in this diagram, your blue and purple spots are going to be your highest density of mussels. And so you can see there's a lot of diversity within that bed, and a lot of it ties to where there's permanent water, and where there's the right flow to keep the gravels clean and to have mussels be in those sites. There are a variety of factors that can affect mussels negatively or positively. And, water chemistry components are a big part of that. If the water temperature is too warm, the mussels will stress. They're easier to pick off to predation, and they may just die from simply the water temperatures being too high. And consequently, if the water temperatures are too low, that can inhibit reproduction. They don't get those cues that it's time for them to go ahead and reproduce. Calcium carbonate, CaCO3, is really important because that's what they're building their shell out of. And if that isn't available in that system, the shells are going to be really thin and they're going to be very fragile. Tie into that, what's the pH of the water? And if it's more acidic, it's going to erode that shell. And we do see that in some places. And of course, because they're aquatic animals, there is an oxygen in that system that's going to be pretty tough for them to survive. From a substrate perspective, we see them mostly in coarse sand and sand gravel mixes, but there are some species that we find a lot in mud or even in loose clay. And so it can vary a lot depending on the river system. We need water velocities fast enough to keep the site clean, but not so energetic as to erode the site or to move the mussels out of the area. Water depth can vary. You can find mussels in as few as a few inches, in as small as a few inches, but in the big rivers, we tend to find them even in 25 and 30 feet of water. And then, of course, they wouldn't be there without the fish host. Almost all of our native species must live on a fish host in the early life stages. And if that fish host isn't available, you won't have mussels there, or you're going to have a mussel population that's dying. So here's a graphic that really kind of highlights that link between the fish and the mussels. And so in this graph, you can see that as we move along the x-axis, the more fish species that we have, the more mussel species that we're going to have in those systems as well. And when we talk about mussels and their hosts, we talk a lot about whether they're generalists or they're specialists. And so generalists are species that can adapt and their young can attach to a wider variety of fish hosts. And so you're going to be more likely to see those down here in these lower numbers. They can adapt and attach to a wider variety of fish hosts. But as you start to increase those number of fish hosts, you have a greater chance that some of these are going to be limited to being only on a specific fish. And there are several species that we do find that in. And oftentimes those are the mussel species that are showing up on the endangered species list because maybe the fish is having a hard time thriving in the system. And so the mussel is suffering because of it. And then we tend to find more diverse mussel communities in places where the river systems are bigger. You've got larger drainages, larger watersheds, and as you move down into the drainage, you start getting into a bigger river system, and we start to see more mussel species and more mussel numbers in those sites. So larger streams will have more taxa, but even some small streams can have some diversity as well. And again, we're looking at, is it a stable stream? Does it have permanent water? And is the fish host present? And even some of our ditches, whether they be natural or constructed, can house a variety of different mussel species, can be very diverse. And we'll talk about one of those specifically here in a few minutes. Now, this slide is one that Steve always likes to throw in for a little bit of fun. And he's got three graphics here that I'll work through. And so in this first one, basically what he's telling you is that he does not find mussels in places where he also finds snapping turtles or cottonmouth snakes. And he always jokes that, well, it could actually happen that way naturally because, snakes and turtles do eat fish, and so they could be eating the host. But what he says really happens is if I'm paddling downriver and I find a water moccasin, I'm not going to sample mussels there. I'm going to move a little farther downstream. This graphic shows that he tends not to find mussels when it's raining. And he jokes that he doesn't really like to work in the rain, but this actually is true, that if we're getting lots of rain, the river systems tend to be muddier, so it's harder to see the mussels. And when they get really flashy, it's not much fun to try to be out there. Then this last graphic shows that there seems to be some sort of association between mussel beds and bridges. And we always talk about that, you know, when the engineers are putting in roads and bridges, they do tend to try to select stable sites. And we find mussels at stable sites, so it makes sense that we might find them around bridges. But in reality, bridges are just great access points for us. It doesn't mean that there aren't mussels in between the bridge crossings. It just means that we've tended to focus on those bridge sites for getting down to the river. There are some mussels that have adapted to be very specialized. And Spectral case is a federally endangered species, but Missouri is a stronghold for, and they are one of those specialists. They're found in these dense aggregations. They like lots of big chunky rock, and oftentimes they are associated with those bluff pools because that large rock falls down off of the bluff, falls down into the river, and creates this habitat for them. Now, sometimes we accidentally create good mussel habitat. And an example is in the range of the spectral case mussel, where state and county highway departments have gone in and put in bridges and then used large chunks of riprap, large chunks of rock to try to armor around those. They create a great spectral case habitat because the spectral cases will use those spaces in between the large rocks. And I had it once described to me as it was like picking your mail out of a mailbox. And I've heard of some cases where divers that were doing work could reach into some of those rocky places and be all the way up to their shoulder and still be feeling many more mussels farther back up in there. Another one that's gotten to be a habitat specialist, but a little bit different in that it has a host specialist as well. This is a salamander mussel. It likes those large rocks as well, but it has adapted to where its host is not a fish, but instead our only true aquatic salamander, mud puppy. And so where you find mud puppies, you're going to find the salamander mussel. And then we have the federally endangered fat pocketbook mussel. It's a species that the stronghold in Missouri is Bell Fountain Ditch, which is down in the Boot Hill area. And this picture shows Missouri on the right-hand side and Arkansas on the left-hand side. And it has very uniform habitat. It's constructed ditch, drainage ditch, and that's the perfect place for fat pocketbook mussels. Okay. So before I get into this next part, I'll give Amy an opportunity here. If there's some questions thus far, I can answer those questions. And if we don't have any, I'll go ahead and move on to the next section.
Sure. Yeah, we had a question. If the relationship of the mussel with the host, if that's parasitic, and at what point does it end and the mussel becomes independent?
Sure. So we sometimes call it parasitic. It is and it isn't. The tiny mussels they don't take any nutrients. They don't really do any benefit from the host. They don't harm the host. So in many ways, they're not really parasitic. They can survive on those hosts, usually for anywhere from 2 to 6 weeks. But the main thing that happens there is those fish, as they're moving around, that's the main dispersal method for trying to get juvenile mussels, getting your genetic material, your juvenile spread to other parts of the river or to other river systems. And so that's how a lot of these populations grow.
Great. And we also had the question, are there any mussels besides the salamander mussel that have hosts other than fish?
Right now, there's none that we are aware of. Over the years, especially for spectral case, for a long time, we didn't know what the host was. And so there were people trying a whole variety of different things. They were trying dragonflies, crayfish, um, hellbenders, I mean, all kinds of different things. And more recently, they've discovered that it is goldeye, I believe, is the primary host for spectral case, and they've been able to get them to transform in a lab. So very unique situation and a long time trying to find out what that one was.
Great. And I think we had a couple of chuckles on why you don't find mussels near where there's cottonmouths and snapping turtles. So I think we kind of understand that one.
I forgot to throw in Steve does a story with that one as well, that there was a time they were doing sampling in a stream in southern Missouri, and somebody had jinxed the scenario and said, wow, we've been going a long time, haven't seen any cottonmouths. Boom, guess what? They saw a cottonmouth. And he said that he normally, if they're snorkeling along, he'll pop up, look around, see where everybody is, make sure everybody's okay. And where everybody had been pretty dispersed around the edges as they were going along initially, after they found the cottonmouth, everybody was sampling down the middle of the stream. So they were kind of staying away from the cottonmouth habitat as well.
Great. Okay, and one more question before we move on. So why do we need a host for immature mussel distribution?
So, it's one of those things that what we hope for, or what the mussels kind of aim for, and whether they realize it or not, is, those fish oftentimes are using habitats that are stable, that are clean, that are using all of those things. And you can imagine, the tiny mussels are so small whenever they're first drop off of that fish, and even smaller whenever they're still in the female mussel, that they could easily be covered by sediment, could easily be lost, and entire broods be lost because of an environmental circumstance. But by getting them on the fish, and as those fish disperse and they drop off at different times, you increase the chances that some of those are going to fall in good sites and grow up to be adult mussels and pass it on to the next generation.
Oh, more questions coming in. All right.
Okay.
Can mussel beds be created by human action?
Yes, in some ways. We're actually doing some work on the Mississippi River where we are trying to create some favorable sites for mussels. There's some work that the Corps of Engineers needs to do to maintain the navigation channel, to keep water in certain places in the river. And so one of the things we've challenged them with is to try to design those projects to be more mussel friendly. Whenever there are times that there's a project that, there just isn't much choice, that mussels are going to have to be moved out of harm's way, they're always looking for sites to go with those. And the thing that I've always tried to encourage is, as if we can create better sites to eventually take those to, then we get better survival and we get more good mussel beds in our river systems. And so we're always looking for opportunities to try to increase mussel habitat.
Great. Okay, one more, and then we can move on.
Okay.
So how do you actually sample to determine who the host fish species is?
So oftentimes it's a trial and error. If you saw Chris Barnhart's presentation here about a week or so ago, Chris showed a variety of different methods that mussels use to try to get their juveniles onto the fish. And so usually what he ends up having to do is he'll bring in fish from the same systems that the mussels were collected from, and he'll keep them in tiny aquariums, and then he will harvest the juvenile mussels, squirt them onto the fish, and then he can look at them under a microscope to see who attached and who's encysted onto that fish. And then he can, with some really fine filters, hold those fish, and as the juvenile mussels exude off of the fish, they will get caught in the filters. And so he has raised thousands of tiny mussels into juveniles and adults by capturing them off of those fish and then raising them up. And so you can always tell. I mean, some of them, the fish will, the mussels will not survive on a particular fish species. And so that's how he finds the answer out. Or the other thing that sometimes can happen is a particular fish, if it's been exposed before, may build up some sort of immunity. And so oftentimes you'll only use them one time before you bring in some different hosts.
Okay. Excellent. Thank you so much. And yes, Chris Barnhart's presentation from last Thursday was recorded. It is posted on our Facebook page, on the Stream Team Facebook page, and also on the Missouri Stream Team YouTube page for viewing by anybody who wants to see it on their own time. And this one will be recorded and will be posted as well. Thank you. All right.
Yep. All right. So for the collection methods and techniques, I'm going to show you some techniques that we use and some that you may be able to use as well. And again, at the end of this section, we're going to talk a little bit about an assignment for you. And you can, it's a voluntary assignment. You can participate if you want to. And so we'll get right into some of those methods. So one of the traditional methods that have been used on our big rivers historically is a mussel braille. And these are called brails, crowfoot bars. And it's basically a long bar, and hanging from it are these series of crowfoot or braille hooks. They'll have 3 or 4 prongs to them. They're not sharp like a fishing hook, but oftentimes they'll either have a hook or they'll be straight. They may have a ball on the end or not. And what you're doing is you're pulling that down the river. The mussels are burrowed into the sediment, but they're still partially above. And as they have gaps, the mussel braille, the hook will get into that gap. The mussel thinks something's trying to get it. It will respond and clamp down. And so then we pull that mussel up out of the substrate. After we run about 100 yards or so, we can bring that up and look and see what kind of mussels we have on there. As you can imagine, it's not overly efficient, but if you do get into some good sites, you can bring up quite a few mussels. And there's a site down by St. Louis on the Merrimack River. We ran about a 10-foot braille down through there, ran it about 100 yards, and I think we had about 40 or 45 mussels on it when we came up. So you pretty quickly start to figure out when you're in a good spot. More modern times, scuba and surface supply to air systems or hookah rigs have been really good for us to use. It allows you to get into some deeper water where certain species of mussels survive better, and it allows you to stay underwater longer, especially when that water is just a little too deep to sample through other methods. Snorkeling, especially in southern Missouri, is probably the most common method. You can spend a lot of time with your face in the water and your snorkel in the air, looking along and either doing tallies or marking stuff on a board and a grease pencil, and just working your way down through there. And what a great way to spend a day is just working around out there and looking at all the different things you can find on the bottom of the river. View scopes or view buckets, you can actually make one of these. We've done it numerous times. They're an effective method as well, especially great if you want to get kids out so they can see the fish, the mussels, the insects, all the different things that are out there. But the one thing that I will tell you is there is a direct relationship between using a view bucket and having a bad back the next day. So use at your own risk. You can do visual searches. If you're shallow enough water and the water is really clear, you can just go out there and you can wade it along and keep looking for those mussels out there. Oftentimes you do have to be aware of glare. You have to be aware of shadows and other things like that too. Polarized sunglasses work really well for this. You can see so much better when you're sampling this way. But part of what you're looking for is, in many cases, those mussel shells will have a section of them that have been eroded away, and that white part really shows up on the bottom of the river. So that's one of the great things that you can look for, but that's just part of the picture that you're looking for. And then there's tactile searches. And in those cases when you're working in shallow streams, but maybe the water clarity isn't that great, then this is a great method. And so I have to use this a lot in my streams in North Missouri. And the water not being clear doesn't mean that you're not going to find much in the way of mussels. I've had some sites up here that I have found really diverse, really dense mussel beds and really small streams simply by getting on my hands and knees and going grubbing around out there. So this method is oftentimes called grubbing or polywogging as well. Steve did add that for this particular site, this is where a tactile search can be really useful. He said there were cows nearby, and he said, "I'm sorry, I'm just not putting my face down in that poop-filled water." So where to look? So part of what you're going to look for when you're going out there and working your way down a river is you can watch for those muskrat middens. So muskrats, whenever they're feeding on mussels, they'll swim out, they'll pick one up, they'll go back to a nice secluded place. And it might be on a fallen log, it might be a flat spot under a root wad, someplace that's secluded where they can feel safe. They'll pry that mussel open, eat the animal that's inside, they'll drop the shell, and then go back for another. And you can find pretty good piles of them in particular locations. My wife was helping with a survey back several years ago, and I believe they found over 100 shells in one little pile where this muskrat had really found a great cache of them. The other thing that's kind of interesting with looking at the muskrat middens is sometimes they find species that's hard for us to find, especially some of those smaller ones. So you can oftentimes add to the diversity of a particular stream by seeing what the muskrats have been feeding on. But if you're not finding those, you can start to look at the river system itself and start looking for some stable sites. So you start looking at the gravel bars, and if there are little island complexes out there, start looking in those, and do you find mussel shells laying on those bars, on those sand and gravel bars? If you do, then there's probably a mussel bed either upstream or adjacent to that site. And so that's a great place to start looking. And you can also look and see how stable that site is. You can see for that upper sandbar, it seems to be fairly new. There's not much vegetation growing on it, so it's probably still moving and developing a lot. It's probably not as stable. But you can see on those on the little island and the other gravel bar farther down, there's some trees growing in it. There's some other vegetation, and those sites seem to be a little more stable. And sure enough, there's a pretty good mussel bed right in that area. We also talk about the bluff pools, and on this bottom left-hand corner of the screen, that's a pretty steep bluff in through there. And where that large rock settles in, you'll find mussels that are buried in between the spaces in there. And they're a little harder sometimes to search for, especially because of the depth of the water, but you can find some interesting species in those places. Okay. And in some cases, places that you might normally just drive by and not pay any attention to turn out to be pretty good spots as well. This is the same photo of Bell Fountain Ditch, and this is a stronghold for the fat pocketbook mussel. And the thing that Steve tells about this particular site is that this ditch has pretty uniform depth from water line to water line. The substrate is all the same, and you can go pretty much anywhere out there and you can find mussels. They can be near shore, out in the middle of the channel, and everywhere in between. So don't ever assume that there isn't anything there until you give it a good look. If you have a site that has some of these large boulders, this is the upper St. Francis River, and boulders create interesting refuges for mussels, because usually immediately downstream of those boulders, right in their shadow, you're going to find a mixture of sediment and sand and gravel and just everything else. And you're either going to find mussels in that zone or along the edges of the boulders, because they can find stable sites, but they still have plenty of water moving by to bring oxygen, to bring food, and to bring reproductive material. In a lot of our southern streams, because they have so much energy going on out in the middle of the channel, we tend not to find very many mussels out in the main channels of those rivers. And so what we look for are some of these little side pockets of habitat, like this fallen log, for instance. In behind it, you're going to find a little finer sediments, a little more foods mixed in, and a little protection from the current, especially when the river comes up a little bit. And so in these kind of places, that's where you're going to find more mussels, in closer to shore in those finer sediments. And that works in northern Missouri as well. I always look around fallen logs. And when we talk about depth, don't ever assume that just because it's a shallow site that it's not going to have mussels. Especially as you're getting into southern Missouri, there are more permanent seeps and small springs that provide year-round water to some of these small side channels. And you can see there's a finer sediment, finer sand in the background there. And Steve said there were several mussels that were buried in that sediment, in that sand in this area. Okay, I'm gonna do this real quick. Be a little bit light back. Okay, so one of the other things that we oftentimes will do is if someone were to bring us a mussel shell and say, okay, what species do I have here? Usually the first question that we're going to ask is, okay, where did you get it from? And part of that is because there are certain species that are common in certain river systems. There are others that don't occur in those river systems at all. And so if we don't readily know based on what we see, we're going to look a little bit about what we would find in that particular river system. So if I wanted to find pistol grip and or maple leaf, there are a lot of different river systems that I could go to and have a good chance of finding them. But there are some species that are kind of confined to certain river systems, or maybe only certain sections of those rivers. Spectacled case and sheepnose are just a couple of those examples. And you can see that there's really only a few places where there's potential to find both of those species. But then there are others yet that are even more rare, and they're confined to really few sites, oftentimes in very small sections of certain rivers. Curtis, pearly mussel, and southern hickory are a couple of examples of those. So if you're going out there and you're looking for mussels, what are you really looking for? What's the site picture that you should focus on? And I've already mentioned the muskrat middens. Again, a great way to start finding things and give you a site picture about what species you should be looking for whenever you're out there. Sometimes you may go out there and you're snorkeling or just wading along, and you'll see that mussel really just sticking up out of the sediment. It may be displaying because it's trying to get its juveniles on a fish host, or maybe it's just up at the top and is in a position where it's just feeding really well. But sometimes you can find trails too. If there's softer sediment there, mussels will move, and you can follow that trail right to the mussel. Even some of the smaller ones, you can do that, but you may also at the end of those trails find some of the burrowing dragonflies. You may find snails, a variety of other things, but you just never know until you dig a little bit right there and see what comes up. Okay, so we talk about having a site picture for finding mussels. And I will tell you that there is one mussel in this picture. Can you find it? So this is a little bit of a trick question. This particular mussel has burrowed into the sediment, and it's kind of hard to find. The only parts that you can see are its siphons sticking up above the bottom of the substrate. So as we start to zoom in on one of them, you can see those right there. And you can see that this mussel has the in-current, S-current siphons open. That's the only part of it that you're going to be able to see. And you can start to see some of the other materials being extruded from that mussel. So that's one of the things that you're looking for. But as Steve likes to describe it, we're looking for a rock amongst a whole bunch of rocks. And especially if that mussel has established itself in a very stable, very favorable site, it'll get plankton, algae, other macroinvertebrates growing on it, and they kind of camouflage it. And so your site picture has to be a little bit different in those systems. Here we've got one with a snail on it. This one's burrowed into the other sediment. You may see these siphons showing. And one of the things that you can watch for with them is sometimes they'll wink at you. And so you may catch movement as you're out there, or if you make a hand swipe, sometimes as they close, they'll squirt water and you see that water shoot out of there, and those mussels are burrowed into those sites. So that's some of what you're looking for. The last little section that he had in here was talking a little bit about the laws and the regulations governing mussels. So if you're going out and you're going to do some sampling, there's a couple of things you need to keep in mind. In Missouri, our wildlife code says you can't do anything unless we tell you it's okay to do it. So you can't molest wildlife, and particular species in particular are protected. So some of the freshwater mussels that we have, some of them are state protected, and many of the ones that are state protected are also federally protected. So we really don't want you going out there and trying to find spectral case or trying to find fat pocketbooks, because we want to let them do their thing in those systems and let those populations rebound. There are some non-native species that are prohibited. We don't want you having in your possession. We don't want you transporting them around. So a lot of folks do know about zebra mussels and quaggas. They attach to many things, rocks, sticks, and native mussels, and they can literally choke them out. And there are numerous cases where people have gone diving, had their wetsuits on, gone diving in areas that were infested with zebra mussels, and they didn't treat their gear before they went to another water body. And when they did, they introduced zebra mussels to those new places. You don't want to be responsible for introducing zebra mussels to some of our really pristine native streams. Chinese mystery snails are a very large snail that's used in the aquarium trade, and more than likely they were introduced into Missouri from people who dumped their aquariums out, didn't want to kill Charlie the mystery snail or whatever. And we have them in several streams down in the Springfield area. Knob-Noster State Park also has them. Again, they get to be fairly large, and interestingly, muskrats love them as well. And then I do not believe that, oh, yeah.
Oh, Travis, I just have a quick question.
Sure.
So when we're out monitoring and that kind of thing, now, if we have these prohibited species in our possession, but they're preserved in alcohol, is that acceptable?
Yeah, I believe so. And typically that's going to be a part of a registered program. So if you're participating in a stream team activity, something like that, and you're going, hey, I think we found Chinese mystery snails down here, we definitely want to know about those, so we know where those snails have expanded to, and having one that we can physically look at is way better than looking at just a picture. So definitely go ahead, drop it in some alcohol, and try to get those samples to us.
Great, thank you.
Yep. And the New Zealand mud snails, I don't believe we have in Missouri yet. It's more common in some of our western streams out in Colorado and et cetera. But that doesn't mean that we can't get them here. And the big risk with them is that they are so prolific, produce so many offspring. And if you're out there and wading around in a stream and you get mud and some of the sediment stuck to your boots and that kind of thing, and you don't clean that off, you don't treat those before you go to another system, you have the risk of introducing at least some juveniles or maybe even some adults into those other systems. So always make sure that you clean up your gear. And it's one of those things that I always tell people, if you're going to be working in one river system for a particular day or even for a series of days, start at the upstream part. If you're doing a search for mussels farther up in the watershed first, and then you're going 5 miles down to do your next sample, work that direction as opposed to starting at the downstream end and working your way up. Because if you do it that way, you have the risk of moving those invasive species farther up in the watershed, and we don't want to have that happen. Okay, in Missouri, you can collect mussels. Typically, it requires a Missouri fishing permit that allows you to collect up to 5 in the aggregate, whether they're dead or alive. And a mussel shell is considered to be 2 valves. So you could collect two halves of the same mussel and do that 5 times, or you could collect 10 different single valves if you were trying to create a collection. Anything beyond that's going to need a collector's permit, and there's a small fee, and that gets reviewed by Steve and several others of us before we would okay those permits. You can collect them by hand net, or pull in line. Season is open all year. Mussels and clams can be used as fishing bait, and they are used for fishing for catfish frequently. You can't collect those protected species that were on that list that we showed earlier. And then Asiatic clams or Asian clams are non-native. Those can be collected and used in any number. We just ask you to not spread them around to other streams. So, although they're probably in about all of our streams anymore. Okay. A couple of federal laws. There's the Endangered Species Act, and a lot of folks do associate this. Usually a species goes on the Endangered Species Act, but that authority, that opportunity allows us to protect the habitats that those animals or those plants occur in. And oftentimes the things that we do to protect the special species are a benefit for all of the other critters that live in that system as well. And so there are lots of positive that happens whenever there's an Endangered Species Act listing, even though it's a species that's kind of taking it on the chin at the moment. And then the Lacey Act is a federal law. If you were to illegally collect species, take species in a state, as soon as you cross state lines, then that becomes a federal offense. And it's punishable by some time in federal prison, higher penalties. And so it's just not something that you want to do. We have used this in some cases in the past on some freshwater mussel cases where people were harvesting them in our southern Missouri streams and then taking them to other states to sell. Okay, presentation's almost over. So this is the point where I'm going to give you your assignment. Okay. Steve is going to be back and doing a presentation on September 16th on mussel diversity and how to identify the mussel shells that you find. Normally, when we do this workshop, we're all in the same room and we bring lots of shells for you to look at and try to identify. It'll be a little harder to do here. So what we want you to do is, if you don't have some native mussel shell already laying around home, go out to a local stream near you, find some freshwater mussel shells, some native stuff, and collect a couple of them and clean them up, hang on to them for Steve's presentation on the 16th. And what he's going to do is he's going to go through all the different characteristics that we look at to try to identify those individual species. And so while you have a shell in your hand, you'll be able to see some of the features that your mussel shell either does or doesn't have as he's going through those. And he's going to show some of the more common species that we find in the state as well. So you may see the exact one that you're trying to identify too. Just as an example, there's got 1, 2, 3, 4, 5, 6, 7, 7 different species of mussels that I have in this photo. And they're all very different, but they all have some similarities as well. So I'll start to wrap things up here. Steve apologized for not being here, had the family matter that he needed to take care of, and it was a very last minute thing. He will be back on September 16th and is looking forward to doing this presentation with you. And then I'll leave this slide up for just a little bit. These are the remaining presentations that are coming up. Andy Roberts with the Fish and Wildlife Service is going to talk about mussel threats and conservation issues on September 12th. Steve's going to do the mussel diversity and identification presentation on the 16th. And on the morning of the 18th, I'm going to do the presentation on our Big River mussel issues, so working on the Mississippi and what that's like. And then I'll also talk about some mussel outreach opportunities, some activities that you can do if you're at a fair or an event, some things you can do with kids, and some very unique online resources that you can tap into that show some really cool stuff related to freshwater mussels. Okay. And from there, since the sun's gone down, I'm going to flip lights on in here. Amy, if there's additional questions, feel free to go ahead and ask.
Sure. Excellent. Thank you so much, Travis. That was an excellent presentation. So I do have a couple of questions that have come in.
Okay.
One was, are rare mussels necessarily considered endangered species? And I think one of the slides may have explained that.
Okay. So there can be, and we see this, there are some mussels that may be rare to Missouri. So they may be considered, rare, threatened, or endangered within the state, but may be common in other places. So it may not necessarily be listed in the Endangered Species Act, but may be listed as endangered within the state. And that happens fairly frequently with some of our species. We may be on the fringe of its range. And oftentimes, if we don't necessarily have the stronghold for that species, we may not get other endangered species funds. Because Missouri is a strong for something like the spectacle case mussel. We have gotten funds in the past to do some of the research on those.
Great. Okay, I have 3 questions. One,
Okay.
is empty shell considered a dead mussel? And
The which
more
one?
than 5, just an empty shell, is that considered a dead mussel?
Correct.
Okay. Do more than 5 in your possession require a permit? And what if it's collected as part of a stream team activity?
Yeah, so I may have to let you define a little more related to the stream team activities. It is one of those things that I think the reason why they set the number at 5 was for a long time we were having some issues. I'll have to back up a little bit here. So some of the historical uses for freshwater mussels in the more modern era has been for the cultured pearl industry. And so there have been places where those good, thick, heavy shells that they would use in that industry were found in some of our interior streams, which were not open for commercial mussel harvest. Historically in Missouri, we only allowed commercial musseling in places where we allowed commercial fishing. So the Missouri, the Mississippi, and the St. Francis rivers were the only places where you could do that. And so consequently, what was happening is there were cases where people were going into our interior streams. They were harvesting large numbers of mussels and having a huge impact. And so the department set that limit fairly low so that folks couldn't use the excuse, oh, well, I'm just collecting fishing bait, because they were just collecting way too many to be used on a particular day. Now, having said that, I can't say that I've ever heard of our conservation agents writing a ticket for someone who had dead mussel shell. I know that there are a lot of places that they have probably gone to and seen mussel shell laying in a flower bed out in front of somebody's house, and there might have been more than 10 valves there. But definitely, if you're out and collecting live critters and keeping them, that's going to raise some flags. If you're out on a gravel bar and you're picking up some old dead shell, they're probably not going to ask too many questions.
Yeah, and as far as stream team activity goes, we do have that one small provision within the wildlife code that if you are for stream team activity like water quality monitoring, it's not necessarily to have a collector's permit for collecting invertebrates during that activity, but you'll be mindful of the possession of what you have based on what permit you have. So if you're collecting crayfish, that kind of thing, you make sure that you still have your fishing permit, as I understand it.
Yeah. Yep.
And I actually had a question myself, because we tell our volunteer water quality monitors when they do collect mussels in their kick net samples to place the valves face down so that they can let the foot come back out. And I just want to make sure that that's correct and that we're providing the right information on that.
Yeah, actually, usually what I end up trying to do is I'll try to put that foot back down in. And what that does is that allows the mussel to stick it out there into the substrate and to kind of dig its way back into the location. Some of them, it's kind of interesting. Some of them do like to be oriented a certain way, and especially at certain times of the year when some of our species are brooding juvenile mussels and they're trying to attract that fish host. In those cases, they'll literally flip themselves completely upside down and will display this outer edge of the shell rather than their top side. So usually I just try to, if I'm replacing them back in, just kind of push that foot end, usually that rounded end down into the substrate a little bit. It won't take them very long and they'll burrow in. And if they don't like the position they're in, trust me, they will change.
All right, excellent. Well, we have a lot of, great presentation. Shout outs from folks from the Lake of the Ozarks Master Naturalist chapter. Just all around, we thank you so much for your time sharing this information with us tonight. So with that, we'll go ahead and close the presentation, but please feel free to contact us if you have any additional questions and join us for the additional presentations that we have coming up on September 12th, 16th, and 18th.
Thanks, everyone, and we'll see you in a few weeks.